Today we'll be talking about an experiment that we conducted for our biology performance task. The aim of our experiment is to see if bread with maltogenic amylase enzymes will have a longer shelf life as compared to bread without added maltogenic amylase enzymes. These are the ingredients that we will be using. Firstly, mix 1 cup of warm milk, 1 tablespoon of sugar, and 2 teaspoons of active dry yeast together. Then let it bloom for 10 minutes until it is foamy. In a separate bowl, mix 3 cups of all-purpose flour, 1/4 cup of sugar, and 1 teaspoon of salt. Then, add 1 large egg, the yeast mixture, 1/3 cup of melted butter, and mix. Knead the dough until it is smooth and elastic. Then split the dough into 2 equal parts and add the maltogenic amylase into one of the parts. Let the dough rise for an hour in 2 different bowls and cover the bowls with a cloth. Roll out the dough to form the shape and place them in a loaf pan with baking paper separating the dough with the maltogenic amylase and the dough without it. Let the dough rise again, then put the loaf pan in a preheated oven for 18 to 20 minutes. After the bread cools, cut out equal pieces of bread and place them in separate Ziploc bags. We obtain our results by using a transparent piece of paper that is divided into squares of dimensions 0.5 cm by 0.5 cm, forming a 15 by 15 square grid. The number of mold-filled squares is then counted and recorded in a table every day by overlaying the bread with the transparent paper of 15 by 15 square grid. From day 1 to day 7, there were no changes to both pieces of bread. However, on day 8, 15 mold-filled squares were observed on the bread without the maltogenic amylase. On day 10, 2 mold-filled squares were observed on the bread with the maltogenic amylase. Moving on, we will be presenting our discussion. Our key findings were that mold was observed on the bread without maltogenic amylase on day 8, while mold was only observed on the bread with maltogenic amylase on day 10. This indicates that adding maltogenic amylase to the bread before baking will help to increase its shelf life. A process called fermentation occurs when all the bread ingredients are mixed together and the dough is left in a warm place. During fermentation, carbon dioxide is produced and trapped as tiny pockets of air within the dough, causing the dough to rise. When the dough is being baked, the carbon dioxide expands, causing the bread to rise further. So, how does amylase help? Amylase is one of the main enzymes that help break down starch to produce reducing sugars like maltose and glucose. These fermentable sugars are used by the microorganisms for their growth and thus help take the fermentation process further. Adding the maltogenic amylase before leaving the dough to rise aids in a faster fermentation process, hence producing more carbon dioxide. Having more carbon dioxide in the dough means that the bread will have more air pockets after baking, which help prevent the bread from growing stale. Less mold would then be formed on the bread for a longer period of time as compared to the bread without maltogenic amylase. Our hypothesis was that bread with added maltogenic amylase enzymes will have a longer shelf life as compared to bread without added maltogenic amylase enzymes. Based on the results, mold took a longer time to form on the bread containing maltogenic amylase, proving our hypothesis to be correct. A practical application is that bakers will be able to use amylase so that the bread can be stored for an extended period of time before it goes bad. This will result in less waste from bread being disposed of. In addition, it reduces the need to replace bread that has spoiled often, reducing the number of trips that bread trucks need to make from bakeries to the stores, thereby reducing the amount of carbon dioxide released into the atmosphere. In this experiment, we only compared bread without maltogenic amylase to bread with maltogenic amylase. Thus, it is unknown how much maltogenic amylase should be added for the shelf life to be the longest. So an area for further study will be comparing the different amounts of maltogenic amylase added to the dough to determine whether adding more or less maltogenic amylase would affect the shelf life of the bread. Thank you for watching our presentation.